Hi, I'm Bryce Ludwig.
I'm Meg Butt.
We're graduate students in the Zondlo lab. Today we're going to be talking to you about some of the projects in our lab that focus around bioorganic chemistry and molecular design. Protein phosphorylation is a major topic of interest in our lab. We seek to understand the functional effects of phosphorylation, as kinases are misregulated in a number of diseases such as cancer and Alzheimer's disease. In studying protein phosphorylation, we found that threonine, upon phosphorylation, undergoes a disorder-to-order transition, which becomes a structural switch within the peptide or protein. We have developed highly fluorinated amino acids, which are used for molecular imaging in 19F NMR or 19F MRI. These amino acids have 9 equivalent fluorines, which exhibit high sensitivity and high signal-to-noise in magnetic resonance. In real time, we can probe phosphorylation or signal transduction. A specific application of phosphorylation which we study is Alzheimer's disease. The tau protein is an intrinsically disordered microtubule-associated protein, which undergoes hyperphosphorylation in Alzheimer's disease and CTE. When hyperphosphorylated, tau dissociates from microtubules, becomes rigid, and aggregates into neurofibrillary tangles. Using synthetic strategies, we are studying the structural and functional effects of post-translational modifications such as phosphorylation on peptides and proteins derived from tau. We have found that phosphorylation and glycosylation have opposing effects on the structure of tau. Our lab uses solid phase peptide synthesis as a strategy to introduce novel post-translational modifications or functionalities into peptides and proteins. This technique is limited in terms of how long you can build your peptide before you lose its synthetic efficiency. To synthesize fully synthetic proteins with defined post-translational modifications, we use a technique called native chemical ligation to ligate 2 synthetic peptides or peptide and protein. This technique, when originally developed, utilizes cysteine at the ligation site. We are developing novel synthetic strategies using other amino acids such as proline or phenylalanine as ligation sites.
Another interesting project that we have is designing of inhibitors of protein-protein interactions. For this, we have designed small molecules that mimic alpha helical recognition motifs of proteins, for example, mimic of tumor suppressor, P53 transcription activation domain, which allow us to suppress the gene activation and gene expression. And the scaffold we have designed is extensible and can be used to develop inhibitors of viruses, including HIV, Ebola, and novel coronaviruses. These scaffold can also be used to design the inhibitors of gene activations in breast cancer and prostate cancer as well. In protein engineering, we are designing small molecules, peptides, and mini proteins that can mimic function of large proteins. We have developed tunable control of protein structure via aromatic interactions for application in nanotechnology and biomaterials. We have also developed novel proteins with high affinity and high specificity to control cell signaling, and we have also developed novel sensors of protein changes in oxidative stress. So we are also working on novel tunable biomaterials, peptides based on nuclear pore complex form, usually stiff hydrogels. So we are exploring the mechanism of nuclear pore transport and developing the biomaterials which can mimic its selective transfer for drug delivery. And we are also working on the novel post-translational modifications such as cysteine sulfonic acid, which is the oxidative post-translational modification and has been found to affect hundreds of identified protein targets and has a few known functions. We are designing novel functional molecules by using synthetic organic chemistry. These modified small molecules can be incorporated into the peptides and proteins to achieve functions which is well beyond those possible with the 20 encoded amino acids. Thank you very much.